Hi, everyone, and welcome to Introduction to Video and Image Analytics for Marine Environments, or VIAME. So really quick, I just want to introduce our speakers and what we're going to be covering in today's workshop. My name is Susie Winquist, and I am a graduate student here at Oregon State University. And I'm going to be just giving a little background on my master's research and my motivation for working with VIAME and provide some context for the work that Aidan has been doing with us this summer. So after that, we're going to hear from Aidan Cox, and he is a research experience for undergraduates intern who's worked with us this summer. And he's going to give us an overview of the workflow he's developed working with the VIAME full frame analysis pipeline. Then we're going to pass it over to Matt Dawkins and Brandon. They're from the VIAME development team, and they're going to be covering just the broader scope of all the functions that VIAME has. And we're going to move into a Q&A session in the second or the later part of this hour. Then part two of this workshop, going to be going through a step-by-step tutorial with the full frame detection pipeline training and applying that to a new video. So a quick introduction to my master's research. I'm using video loggers to study the foraging ecology of breeding Adelie penguins. My advisors are Drs. Katie Duggar and Rachel Orban, and I'm based here at the Hatfield Marine Science Center in the Seabird Oceanography Lab. This project is part of an ongoing research effort studying Adelies in the Ross Sea with funding provided by the National Science Foundation. And our research takes place at Cape Crozier, which is on Ross Island in Antarctica. And I'm focusing on the period of breeding when the adults have larger chicks and they're spending 1 to 2 days foraging in the adjacent Ross Sea at the time that this photo was taken. So for this work, we are deploying both video loggers and accelerometers on the breeding adults, and tags are attached for a single foraging trip and then retrieved. And the video loggers are programmed with a delayed start time, and they record about 5 hours of video during the foraging period, while the accelerometers are capturing the entire foraging period. So my goal with the video data is to first to look at the foraging captures that occurred during the video period and use those to validate prey capture movement signals that we see in the accelerometer data. My goal with the video loggers is to examine the allocation of time the penguins are spending underwater, and then the habitat and behavioral context for prey capture rates. So this second portion is really what's motivated my interest in using VIAME. So it's really easy to generate a lot of video data quickly. I currently have about 56 hours of video from a pilot study done a few years ago, and I'm planning to collect at least as much again this year. So I started looking for a tool that would help me rapidly hone in on sections of the video that were relevant to my area of particular interest. I had VIAME recommended to me by several people, and I started checking out their web-based platform, which is free and has a user-friendly interface, which if you stick around for the second part of this webinar, we're going to have a tutorial in that web-based interface. So this seemed like a great tool to get from just the entire video data and start filtering out some of the areas that I'm particularly interested in, and building an automated pathway to do that. And so this is about the time that Aidan joined the project. And so I'm going to pass this over to him now and let him talk about the workflow that he's developed this summer working with us, and walk us through the process he's been using and kind of the results of where we are so far. And then there's going to be time at the end of this hour for questions. Okay, to you, Aidan.
Hello, everyone. Thank you for that introduction, Susie. Once again, my name is Aidan Cox, and I'm a National Science Foundation research experience for undergraduates intern in the Seabird Oceanography Lab, working specifically over the summer to develop an automated analysis pipeline in VIAME. And before we get started talking about VIAME, one of the things that I just wanted to highlight that Susie mentioned briefly here, next slide, please, is just the size of our data set. So like Susie said, we're planning to collect over 100 hours of video footage. And in the process of analyzing this footage, we're considering the videos at the scale of individual frames. On this slide, you can see over 300 of those frames from a variety of our different videos, which represents only 10 seconds of video at the native resolution of our video loggers. So it is fair to say that we have quite a large data set, like most video and image data. And that's part of the justification for using VIAME to develop an automated analysis pipeline. However, it also presents some challenges. Next, please. Between highly variable images, next, and a highly imbalanced data set where a few categories make up the vast majority of our data set. Sorry. So we quickly realized that between these two issues, the behaviors that we were actually interested in made up a vanishingly small portion of our once very large data set. And so this issue of imbalance quickly became one of the biggest challenges that we faced throughout the process of our analysis framework. And so in the next segment of this presentation, as I walk you through that analysis framework, I'm going to return to this issue of imbalance and try and highlight some of the smaller issues and challenges it created for us and how our lab decided to address those. Next slide, please. So the very first step in our analysis framework was to develop an ethogram. And for those of you who aren't familiar with the term, an ethogram is simply a list of observable behaviors or characters. And this list became the foundation of our later video annotations. And so we identified it as the first and foundational step in our analysis framework. Next, please. So the first step in creating an ethogram is simply to select what categories or behaviors you wish to include. Next, please. And it's here that we run into the first pitfall in our analysis, which is creating too many categories. And this is an issue because too many categories quickly increases the amount of work needed to be done by the researcher beyond what is reasonable. And it has the potential to exacerbate existing imbalances within a data set. For example, we were initially tempted in our group to create a pipeline which could identify individual species of penguin prey. However, we realized that to do so would require a monumental effort to comb through the data set and find enough examples of each prey type to build a successful model. Instead, we decided to consolidate all prey type into a single category to stay true to our research question while keeping the workload to a manageable level. A successful ethogram is also predicated on clear category descriptions. And this is important because clear descriptions inevitably make your later annotations more accurate and precise. This is a benefit to both the researcher, reducing the amount of time it takes to make annotations, and also a benefit to the machine learning process, since computers are inherently very logical. We also recommend creating a photo log of your ethogram, as we found that this inevitably makes it easier for the researcher when you encounter instances which are difficult to classify by the bounds of your ethogram. And we found this to be particularly true when trying to categorize continuous variables such as light level. Once you have a robust ethogram, the next step in the framework is to compile a set of training videos which contain annotations or examples of each category within your ethogram. And here I just want to highlight that while in our analysis, we're focusing on breaking our videos down to the scale of individual frames, VIAME does offer the features and utility to create annotations at different scales. Next, please. Yeah, so no matter how you choose to make your annotations, the first step in compiling a training set is just to select the videos that you wish to include. And it's here that we encounter the next pitfall in our analysis, which is identifying enough examples of rare or transient categories or behaviors. And perhaps the best way it would seem to compile a training set is just to randomly select videos from your overall data set. But in doing this random sample, you might not get enough examples of rare events. And so to combat this, we initially compiled a training set following a more stratified approach by combing through our overall data set to find short video clips containing each category of our ethogram. Sorry, just waiting for the slides to catch up. I think there's 2 more before we're there. Thank you very much. And so once we had these class clips, we were ready for the next step, which was just to upload and begin annotating these videos manually, which sounds pretty simple and straightforward. But I just want to highlight that it's at this stage that organization is particularly critical. Placing your training set in one consistent location and recording where in the overall data set your training clips come from is particularly important in case you need to exclude those training clips from your later analysis. So we recommend taking detailed notes at this step. Next, please. Thanks. So once you have a training set, you're ready to start building an analysis pipeline. This is a relatively easy and straightforward process thanks to the user-friendly interface in VIAME. We'll cover it a little bit more in the dedicated tutorial in the second half of today's webinar. Right now, I want to focus on the next step in the process, which is evaluating the success of your models. In our lab, we chose to do this by randomly selecting video clips and then applying our analysis pipeline to them and changing any incorrect annotations made by these initial pipelines. For our early models, which were relatively inaccurate, we found that the most efficient way to do this was by simply taking the proportion of frames, which we had to correct, over the total number of frames in a video. And I refer to this as the bulk annotation method for evaluating success because it considers annotations from all categories in bulk. And this is great because it makes evaluation very easy and quick, but it also has the potential to introduce another pitfall, which is cryptic failures. And what I mean by that is the bulk annotation method is essentially taking an average of the model's success at predicting each category. So if there's high variability in the model's performance across different categories, the bulk annotation method won't reflect this. And so for our models, which were more accurate and required fewer annotations, we decided to switch to confusion matrices to track model success. And confusion matrices track the frequency of predicted categories relative to the frequency of correct categories within a given video, such that the cells along the diagonal of the matrix represent correct predictions by the model, while the cells in the periphery, here represented in blue, represent different types of error made by the model. These matrices allow researchers to relatively easily track the success of models at predicting events in each category. However, we found that they were particularly time-consuming to create and maintain. And so we only used them in the later stages of our analysis process when our models were greater than 70% accurate. Next slide, please. Well, I can hear our motion sensors. So while our initial models weren't particularly accurate, following this analysis framework, it was relatively easy to employ an iterative cycle of model development. And what I mean by that is, as we saw in the last slide, when evaluating model success and manually correcting annotations made by our pipelines, we were essentially creating brand new training data, which we could incorporate to address some of the common mistakes made by our models. So by feeding these corrected annotations back into our original training data, we could relatively quickly build more and more accurate models. And we just finished the third round of this iterative cycle and are up to about 27,000 manual annotations spread across our various classes. You can see the frequency of those annotations broken down by class here in the table on the left. And what you may notice right away is that between the most common category, open water, and the least common category, bioluminescence, spans 3 orders of magnitude in frequency, which just goes to highlight the pervasive imbalance in our data set. And for those categories which have several thousand annotations, we're noticing that the model is consistently above 80% accurate. However, for categories which have substantially fewer annotations, we can see similar levels of accuracy in categories that are particularly easy to distinguish, such as overexposed images or under ice foraging. The flip side of this is that for categories which are particularly difficult to identify, we're seeing intermediate levels of accuracy even after thousands of annotations. This is the current issue that we're facing in our analysis process. And in consulting other researchers, we believe that we might be able to address it with even fewer annotations by creating a more balanced training set. Next slide, please. And so in summary, large data sets can be both a blessing and a curse, particularly when they're imbalanced. And in our analysis framework, we recommend addressing this problem at 3 particular stages: developing a robust ethogram, creating a balanced training set, and strategically evaluating the success of your model, particularly at different categories. And by following this framework, we found that it was particularly easy to develop an iterative cycle of model development, which allowed us to quickly generate accurate models using VIAME. With that, thank you very much for your attention and patience. I'd like to now give the floor over to Matt and Brandon, senior developers of VIAME, who can give us a little more specific perspective about their software. Matt and Brandon, thank you so much for joining us today. The stage is now yours.
All right, just bring up my slide deck here. Okay, hopefully everyone can see my screen. Okay.
Yeah, looks great.
That's perfect. So I guess thanks for inviting me here. As Aidan and Suzanne mentioned, my name is Matt Dawkins. I'm a software engineer at Kitware, a small software company headquartered out of upstate New York. I'm also joined by Brandon Davis, who will be presenting in a moment, also from Kitware, who's one of the principal engineers on the web application that they've been using for some of this work. If you've seen any of our previous presentations, this might be a little bit of a repeat for you, so sorry for that. These are sort of some generic VIAME overview slides that we typically do to people who haven't encountered it too much before. But first off, what is VIAME? Over time, it's essentially evolved into a do-it-yourself toolkit that biologists, or really anyone, can use to generate their own machine learning models with little or no programming experience. Now either in web or desktop interfaces or command line tools. Additionally, we have recently added an online data repository for storing imagery and related annotations across a few different efforts that I'll highlight in a minute at viame.kitware.com. Most of its current use so far has been in the first 3 items of the below figure, particularly object detection and tracking, but more recently we've been expanding its capabilities into stereo measurement, image enhancement, image registration, low shot learning, 3D model generation, and a couple other fields. Lastly, it provides a few tools to assist with evaluating performance of different algorithms, both while generating models and after the fact. In both sort of binary and source format, VIAME is released open source with a permissive license, so it's all open source. Almost everything we do in this domain, we try to open source where we can. Sometimes a customer comes along who says, we don't want you to open source that particular model, which is okay. But for the most part, everything we're doing is free to use. Some of the things we've been doing are pretty generic purpose. They don't have to be applied just to marine biology. They could be applied to anything, really, any problem. Others are very specialized for individual problems that we've encountered. So just to give you a feel for some of the data that people have been working with, it's a diverse range with a lot of different problems and challenges. Shown here are images from downward facing cameras pointed at the seafloor. This is really how I got involved in these projects initially. Many of these problems require their own illumination just because they're underwater, under a fair amount of depth pointed at the seafloor. So these images, for example, from the HabCam in the Northeast, Northwest Atlantic rather, but Northeast US, an AUV in the US Northwest, I think off the coast of Seattle and lower Alaska, scallop images from the UK. Many of the problems here are going after commercially fish species like scallops, but other are looking for bycatch or invasive organisms, such as in the mid right there. Aerial surveys are also fairly popular and something we do a bit of at Kitware. With most fishery science centers across the NOAA having pinniped detection programs from either manned fixed wings or small drones. So you can see a diverse range of different climates here with different types of seals and sea lions in the top, all the way to the Arctic and warmer climates in the middle, for example, off the coast of San Diego. Scene segmentation has also come up with a few groups recently, for example, classifying land cover. Lastly, we've been seeing some people try to detect some other things from the air, such as sharks or penguin colonies more recently. Outward-facing cameras are a bit more diverse than the others, with a large range of different sensors, color, grayscale, targeting fish populations around the world. Checking objects here typically becomes more important to prevent duplicate counting the fish or whatever we're looking at. It depends on the applications. These might be underwater onboard fishing vessels for electronic monitoring, or just cameras strapped to penguins' heads. All these interesting applications I didn't really know existed until working with NOAA and some other partners. So just to give you a feel for the assortments of platforms collecting this imagery, there's a large variety. Some are towed vehicles with fixed cable to the surface. Others are more autonomous. Aerial, we typically see small UAVs. People are wanting to collect data and even start to do onboard processing on quadcopters. Traditionally, manned fixed-wing planes has been the most prominent here. Planning for VIAME really began in late 2015 with the formation of NOAA's AISI committee. I think development picked up a bit more around 2016, 2017. AISI was created with a representative from each fisheries science center around the US, then also a couple of members of industry and university partners. VIAME wasn't the only product funded by AISI. Others include CoralNet, UC San Diego, David Kriegman, an open data store for coral annotations, then some specialized applications from SRI and Lenel. I guess in 2016, the 6 NOAA members of this committee were awarded a Department of Commerce Gold Medal Award for funding some of these things, CoralNet, VIAME, et cetera. So this committee is how Kitware got involved. For those of you not familiar with Kitware, we make open source software in a few areas with our 4 major departments being shown here. Being open source, most of the software is free to use and available on sites like GitHub for download in source and binary formats. Some of our more well-known software is CMake, VTK, and ParaView. Anyone who's built other computer vision image analysis toolkits like OpenCV or PyTorch has probably encountered CMake, but they're used for cross-platform builds. VTK and ParaView are used for visualization, heavily at government labs, but a bit outside of that too. We're headquartered in upstate New York, but have offices in North Carolina, DC, New Mexico, and Lyon, France. The history of VIAME, basically due to multiple organizations across NOAA having different implementations of the same components, for example, detection or color correction. One of the original goals of VIAME was actually to function as an integration platform, more so than what it evolved into. There were base classes for different things like detectors, image readers, reading classes for reading different formats for things. And this was coded in C++, mostly to encourage use as for optimized embedded platforms as well. but also so that you can write derived classes either in Python or C++, Python being fairly popular with a lot of machine learning developers these days. I won't really go into this too much, but if people have questions at the end, feel free to bring it up. What it actually evolved into over time, though, was basically a do-it-yourself toolkit that people can use to generate their own models. So interfaces connected with the algorithms, doing a couple different things. So for end users, there's currently 3 workflows for generating models for new species or objects of interest that don't exist. The more traditional approach is the middle. The user has some raw data, they do a lot of manual annotations on them, then train a model. And by annotations here, I might mean one of several things. I might mean for object detection problems, the box around the object, its category label. For full frame problems, that might just be a box around the entire image. The second workflow is when you already have an existing model more similar to what you're trying to do, either from someone else's work or for like a first pass that you've trained up on your data. In these cases, it might be more helpful to correct the output of an existing, detect or run some new data as opposed to doing them from scratch. But that's very problem dependent. Sometimes that speeds things up, sometimes it doesn't. The same workflow for updating those models is then used once you have new annotations. So the last workflow on the right is a bit different. That I'll demo in a couple slides, and involves learning from few training samples from a single exemplar as opposed to many, which is what traditional deep learning uses. We're talking about thousands of annotations per category or higher. So there's different types of annotations that pretty much all the annotation interfaces and model trainers support. Box level for detection problems is most common. Say you're counting the number of organisms on a particular image, simply because they're easy to get annotations for. Frame level makes sense in a few circumstances, such as going frame level properties like substrate background, or in the case of these penguin cams, if you're just going for what activity is going on. Key point annotations are most useful for measurement problems, though some people do detection by them as well. And pixel level annotations are harder to generate, but they typically improve detection performance by a couple percentage points. So over time, VIAME has basically evolved to have a couple different interfaces. The newest Dive is sort of shown on the top video here. That can function basically in the web or on desktop. That was one of our motivations for making it. Brandon will show a little bit more of that in a second. The second search is basically an optimized tool for rapid model generation. So given just a few exemplars of a target, searching a video archive for additional samples, quickly generating a model. Generally, it's good as a first pass on the data. Lastly, there's View, our older annotation tool, but it's still in VIAME, and Seal for multimodal support. So optical and infrared at the same time. That's sort of a specialized application. So these 3 interfaces from the user perspective, as you get lower and lower into basically the desktop applications and command line tools, sort of the amount of customability of the algorithms change, but the ease of use for the algorithms from the user perspective also goes down because it's more complex, it's less documented, et cetera. For people coming into the system for the first time, the web application is usually the easiest because you don't have to install anything. You just go to a website, you upload your data, you do some annotations, et cetera. The desktop apps, going a step down, do have slightly more functionality right now than the web server. Typically what we'll do is we'll code things up on the algorithm side first, and then we'll make the interfaces around them, user interfaces. So if you go down to the low level, there's a couple extra features that aren't in sort of the high level interfaces yet. As an alternative to training models in graphical interfaces, there's also project folders and examples within VIAME. Project folders are typically when you just take your images and video, you have some annotation CSV or JSON file alongside that, you put it in a directory, and then you double click a batch script to run training on it. That's its main use case, but from project folders, you can also launch basically graphical interfaces. Examples are sort of used for functionality that aren't in any of the graphical interfaces GUIs exposed yet. So there might be something, for example, say a complex stereo measurement pipeline that might not be loadable into interfaces and runnable yet, but you can run them in these example in the default install. So I'm now going to very quickly go through each algorithm component in a little bit more detail, but not heavily. If there's any questions at the end, feel free to ask, but I'm going to brush through these slides fairly quickly. On the object detection front, one of the first things we did was integrate things from the wider computer vision and image analysis community. So things like fast RCN, YOLO, sort of default detectors that people in the community tend to know. But secondly, on top of that, we've been adding a couple extra things. One is a framework that we came up with for training models that basically does automatic hyperparameter tuning to try to ease the burden on biologists of setting some settings for training models. If you give it enough time and you turn on enough features, it'll run multiple model training at once and then try to choose the best one. But then also things like ensemble classifiers, so taking multiple detectors, running them alongside each other, fusing the results into a single result for output, say on the detection problem on scallops. And auxiliary data fusion, for example, taking like motion and combining that with standard RGB, so you're not just training on a single frame standard RGB. Scoring tools, there's a couple of them also across the system. That's a good example of something that we don't have in the web GUI yet, but is in some of the desktop example folders where you can go and take a computed detection file and put that alongside a ground truth file for scoring the output. Interactive search and model generation, I'll very quickly show, but it's basically a way to do image search on an archive. So given one example of what you're looking for example, here we'll show this flatfish. I don't know what type it is. I think it's a I don't know what it is. But here the user basically presented a single query example. The system gave some returns over an archive of what was most similar to that query sample, and the user can then go through, mark off what was right, what was wrong, create a classifier in this fashion. So it's a relatively easy problem because the fish was very distinct, but in harder problems, it becomes much more difficult. This column on the bottom here are not the top results. They're things that are closest to the decision boundary of the classifier. So it's things the system is more uncertain about. You'll see things like rocks that are the same time, the same size as flounder. Most of the time, if you're able to generate thousands of annotations per category, this approach will be worse than a standard deep learning model train. But if you have very few annotations per category, either because you don't have the time or you just don't have the data, this approach can do better for those few category samples. So just quickly rolling through 4 of our sort of other features. One of the things we've been working on with Conamessa Farm Foundation is coming up with stereo depth maps, basically from pre-calibrated cameras, fusing those into detections for improved performance of buried things like flounder, flatfish. Registration and mosaicing comes a bit more in the aerial domain as opposed to underwater. Sometimes it can come up in underwater if you want to, say, perform a registration-based tracker as opposed to an appearance-based tracker to not duplicate count images or species of things between frames. Typically, in most of our aerial problems, we only have like 20% overlap between neighboring frames. So registration might become more important there for not duplicate counting certain sea lions and seals and things like that. Also for cross-registering different modalities, so for taking infrared and aligning it with thermal, for example. Object tracking, is one of those things where we typically have 4 or 5 different object trackers based on the domain. For example, our high frame rate fish tracker might be very different from our low frame rate aerial tracker. Additionally, we've been adding basically tools to help speed up the annotation process. For example, the user draws one box on a target, runs a tracking pipeline, and then it fills in the remaining boxes. Stereo measurement might not be of interest to this community, but similar to some of the other things, we typically have 2 approaches. We have one for when you don't have many annotations using non-deep learning approaches, and one for when you do have lots of annotations. We've been training up these models for the first time the past 2 weeks because we just got a large annotation drop on Zoo Universe for fish head tail points. So we're sort of just seeing how these do for the first time. The deep learning methods, method 1 has been in the system a while. Lastly, we have full frame classification, sort of similar to the other problems. We have SVM models that are trained, that can be trained when you have fewer annotations per category, and then deep learning models for when you have many categories. Right now, they're both using fairly common techniques in the vision field. So the ResNet 50 approach is just a standard classification convolutional neural net. That's sort of the default method for the deep learning method. And then the SVM method is sort of similar. It's taking a similar ResNet 50 model. It's just trained on a lot of other objects. So on a standard problem called ImageNet, where you have thousands of categories of people and vehicles and things like that. But it's not taking that network as is. It's slicing off the tail end of that network, the classification network component, and just using earlier layers and using a support vector machine to do linear classification on those earlier layers, which is, still a machine learning model. It just has significantly less weights than a default convolutional neural network, which means it'll overfit less to a smaller number of samples. But if you are able to get lots of samples per category, generally, things like ResNet will outperform the prior. Lastly, most people are fine, doing their data collections, bringing their data back to the lab, doing processing on their servers, the beefy servers at the lab. Some people don't want to do that. They want results immediately, either downstream from the camera, because that's going to control what type of data is collected, or midstream, maybe it's not directly on the camera system itself, but it's on the plane, such as the case here. You might have just a server connected up to cameras. We've been doing a bit of work there. So current and future work, one of the near-term goals is just retraining some of the default utility pipelines for fish and some of these other things to help people speed up annotation. The web GUI is currently going through, a couple additional features we're adding in there. Some of the things in like the examples folder that don't exist in the web GUI yet. We've been doing a lot of development. KitWare also does a bit of work with other government organizations. So we've been able to sort of make use of cross-funding from some of our other efforts here. Lastly, is just some additional algorithm specializations. For example, full frame, I could foresee us doing a bit with down the line. There's a lot of things you can do with temporal information and comparisons there. Pixel level classification is something we've been working on the past couple months for the first time. We now have a pipeline that turns basically boxes into polygons, but we've been iterating over the exact pipelines and algorithms that we've been using there. Lastly, expanding into some new domains such as acoustic data processing, electronic monitoring, et cetera. So I guess I just want to thank NOAA, for helping fund this, other organizations like CFF. With that, I'll pass it off to Brandon for a quick dive interface summary.
Cool, thanks, Matt. Let me get set up real quick.
Get myself off.
Okay. Could someone just give me like a audible, how good is size? A little too small, about right?
That looks perfect.
Okay, yeah.
I'll make it a little bigger. So I am just going to give a very brief overview of sort of what the different parts of this interface are, and sort of how you can use it. I assume that most of this workshop is going to focus on the web version, and so something that I would like to point out, I've got this meeting thing up here. The first thing that I'd like to point out is the documentation. And so you can go to, once you've created an account at viame.kitware.com, a button to open the user guide is right there in the top right-hand corner. It'll launch this page. You'll notice this is the dive documentation, and you'll see try the web version and get the desktop app. The desktop app refers to just the desktop version of this particular interface. This is just one of several interfaces in the VIAME toolkit, suite of tools. Again, I think we're going to be focusing on the web version for this workshop, but just to mention, if you pop down to the desktop version, you can get to the full VIAME toolkit installation at VIAME/VIAME. There are all of these installers, and the Windows and Ubuntu both come with the Dive desktop installation. If you go over to the web version, you can click on Try Our Public Server, which is just a link to viame.kitware.com. And again, just briefly, I'm going to be giving some like housekeeping information about how sort of data works on this system. So once you've logged in, you get this page. This is your personal workspace, and you should probably just see two folders to start out with, your public and your private. Data that goes into public is visible to anybody with an account. It's just visible, it's not editable. Data in your private is only visible and editable to you. Something that most people inevitably want to do is to share some data with someone else. I've got this workshop directory that I've set up myself, and you can share data that you've uploaded by right-clicking, going to access control, and finding someone else on the system. I'm going to share this data with Matt. I'm going to give him edit access, and always click include subfolders, and I'll save that. That's just something to keep in mind when you do get around to wanting to collaborate with other folks. Something else that you can do in terms of like discovering data that we already have, if you go to this globe icon and over to collections, you will probably see this training data collection, which has all of about 1.6 terabytes of all sorts of data that we already have. Underwater data, all sorts of other kinds of data. I've also got a workshop folder here for a workshop that we did with NOAA not too long ago, if you'd like some smaller bite-sized stuff, some introductory stuff. And if you're just looking to get started, what you can do is any of these folders that have a launch annotator button, those are data sets, and those can be cloned. So you can make yourself a copy of any public data on the system. It'll launch a window like this, and you can go into, for example, pick a folder to drop it into, clone that data for yourself, and get yourself an editable copy of, again, any public data that we've got. One of the last things that you might want to do is like download data that you've annotated or found somewhere else, so you can get your image sequences, detections, etc. In terms of model generation, just to point out where, after you have run your training or pipelines that I'm sure this group is going to get to later, once you have run a training, you'll get this VIAME folder in your personal workspace. There will be this VIAME training results folder. And so all of the models that you train are going to get dropped into here. And those will be visible once you go, for example, if you have, I'll go back to my workshop, data without annotations. So if you select one of your data sets, you can run training on a group of data sets, but you can, you can run a pipeline, and anything that you train will be over here in the train sub option. And I will not get too deep into this. I'll let the folks running the workshop cover the specifics for this audience. That is just about all that I wanted to cover. So at this point, I think we can probably open it up to questions. And I won't stop sharing yet unless there are questions, in case there are questions that require visual aid.
Excellent. Thank you very much, Matt and Brandon. For our participants, you're welcome to unmute yourself and ask your questions, or type your questions in the chat, and we will kind of serve as the moderators for this portion and kind of facilitate whatever questions you have.
Hey, Aiden, Moritz here. Hey,
Hi.
say, when you train a model, which model accuracy metrics can you get? Can you get F1 score and such, for instance?
I'm sorry, Moritz, could you repeat the question? I didn't quite understand.
Oh, sorry. If I'm breaking up, yeah, my internet is not great. But
No worries.
in the model metrics, when you run one, can you get different metrics such as the F1 score, or what does it compute?
So I can take that one. Currently in the web interface, there's no capability to actually evaluate the models. It's only on desktop right now.
Okay.
What we've traditionally had, but you can on desktop. What we've traditionally done is ROCs, so receiver operating characteristic curves.
Okay.
We have precision on one axis, false alarm on the other. Then a lot of fixed metrics for tracks, object tracks, which is a whole different ballgame as opposed to detections or full frame classifications when you're talking about temporal accuracy and accuracy of linking. So there's a separate tool in there for scoring track performance that measures things like tracking precision, fragmentation, percent detection at the track and detection level, which is basically just fixed metrics at a particular operating point. So you give it a confidence value to evaluate things at. In terms of F1 scores in the current release, we don't, but we're adding a couple new scoring things very shortly that has basically precision recall curves, not just ROC curves and things like F1 as well, area under the curve. So it should spit out some of those things soon. Our training API and evaluation API is the best. Right now, it just functions off of a single detection and ground truth file, so you can really only run it on like a single sequence at once.
Yeah.
We have to do a little bit more to generalize it better, but I guess the key thing is that all the interfaces you have access to the raw detections. You can download a CSV or shortly JSON file that has all the scores and information and localizations in it, so that people can run their own sort of analytics on it. But
Right. I have a follow-up question, but it's fine if Grant Ballard goes for anybody else. I can come back, whatever works best for you.
That sounds great. Thank you, Moritz. Grant, I think you're up next.
All right. Thank you. And thanks for doing this. This is really, really useful. I had a question specifically about the way you're doing the categories, and basically, is there any sense of sort of hierarchy among the classes when you're classifying frames, or can frames have more than one category? Because obviously you would want, for example, you had Pingu head, which is like a frame that has more than 50% of the penguin. It's filled with 50% or more of the penguin's head, but you could still theoretically have identifiable prey in that frame. So I would think you would want to prioritize prey in that case. So can you say a little bit about how that works?
Yeah, absolutely. So for the single frame annotations that we've been working with, each frame kind of has one box annotation drawn around it. And so you can only do one annotation per frame. So yeah, that's part of the reason why I say clear descriptions are particularly important, because we ran into a lot of cases where we had to choose between two different frames. And generally, based on our experience, you're absolutely right. I think it's better to prioritize frames that have fewer training examples, just to kind of beef up those categories and reduce some of the imbalance in the data set. But for some of the other annotation types that Matt was talking about, like box annotations or pixel annotations, you can create multiple of those in a single frame. So that kind of mutual exclusivity is unique to the single frame classification.
Yeah, from the VIAME perspective, I would add, I guess, two notes to that. One is there's now, there's generally, with each of these core annotation types, there's one core annotation like type, but then there's also what you can make arbitrary attributes that's in addition to that type. If you press the A key, or there's an arrow key on the dive interface where you can switch between sort of the core type and one where you do have multiple listings of arbitrary per box as well. So you don't necessarily have to make a second box. You could just assign multiple attributes to the same box. This problem has come up a lot on all these species identification problems. Most of the algorithms we're using by default under the hood, they don't use hierarchical information. So we generally recommend people do the lowest level of the category But then in the desktop version of the software and project folders, you can sort of merge your categories together into the same super category, and you do that after the fact. And we'll add that in Web 2 eventually, where say you annotate 10 types of fish that are really close, but you just want 2 types of like genus level as opposed to species level. You can still do that in the model training tool if you just make a line that's like the 5 species of fish that are one genus, and the next line that's the 5 species of fish in another genus to train a multi-tiered model. And if you annotate the finest level of annotations, you can still go back and do that after the fact relatively easily because you're just merging these 2 categories together. But if you don't do that in your initial annotation, and you want to do some finer grained level annotation later, you might have to go back and relabel your data. So generally what we've been recommending is people do, the finest grain they might want to detect, and then they could go back and do like super categories later at train time.
So thank you for, yep. Grant, does that answer your question?
Yes, thanks.
Okay, perfect. It looks like the next question we have came in through the chat. It comes from Brendan Chasko. He says, I have never dealt with data sets this large before. Are cloning or GitHub issues that I should be aware of? Will I need special permissions or a special account? From our side of things, we haven't had to implement GitHub yet. We have instead been using like physical hard drives to pass back and forth, which is a feasible option since our team is located in the same location. But GitHub could be a valuable tool for collaborating specifically with people in multiple different locations. But I wouldn't say that it's a requirement to process large data sets using VIAME or other tools. And as far as I know, you don't need any special permissions to upload data from different sources to VIAME.
Yeah, I think the we have this public viame.kitware.com where it's a fairly big archive. It's 15 terabytes. But if people get to the point where they have like 10 terabytes of their own data, we wouldn't recommend using our viame.kitware.com. We would recommend they spool up their own server. Just like the desktop application, the web service is also available as a Docker container, so people can buy their own server hardware, hard drives, whatever, and then host a web server locally, either on the internet or on their own local area network. So if you are talking about terabytes of data, that's usually a good way to go. We haven't instituted any limits on the amount of data that people can post on viame.kitware.com yet, because we haven't been approaching the max data limit yet. But if we were to, that's something we would start cracking down on people. It's not a super hefty server. It's more or less just a I think it was like a $5,000 to $10,000 server with about 2 GPUs in it, a 15 terabyte RAID away. So it is backed up, but it's not super huge.
Thank you, Matt. Scott, I think you're next in the queue.
Yeah, thanks. Just very general question for a beginner. So if we had the opportunity to have a really large training set, but there's a huge range of quality, blurry fish, pixelated images, dark, etc., contrasting backgrounds, is there any general guidance about how desirable it is to get a lot of poor quality samples in your training set, or do you specifically want to exclude those, or how do you approach that issue?
Yeah, so this is an issue that we faced as well early on. Initially, when I started on the project, our approach was to go through a video and only annotate the frames that were of like reasonable quality that could be identified. And one issue that we ran into almost immediately was that when training our pipelines, those unannotated frames in the video became a new class in the model that we needed to get rid of because it was popping up as everything. But so yeah, if you're doing single frame annotations, it's important to note that each frame in a video clip must have an annotation. So if you wanted to kind of comb through your data set and filter by the quality of the images, you would have to do that before you uploaded the data into VIAME. So it was all a single contiguous clip, which we found wasn't really feasible. And so we just annotated everything we came across.
Matt, I'd like your clarification. Actually, you go ahead.
Yeah, so I was going to say, a lot depends on what you actually want to run on your final video sequences. Do you want to run on videos of arbitrary quality, or do you want to only run your resultant algorithms on, the good quality ones and see what they are? I think there's a couple of ways you could go about handling quality. One is you can make a classifier, a full frame classifier that's like, is this video or imagery good quality on the current frame? And then you could predicate doing your other stuff on top of that. So you could make an object label class that's like bad quality video and bake that into your classifier. And then it'll classify frames as bad or good. But a lot depends on what you want to actually see as the output from these algorithms. Do you want to see just when the image quality is bad and you can't tell one label called bad imagery, or do you want to see the algorithm make its best chance guess against that? I mean, if you can see that there's discernibly one class in there, there's a decent chance that the algorithm can learn that class as well if there's enough samples of both good quality and bad. But if you really can't see and you don't think the computer is going to be able to know that just because the image data isn't there, these things aren't magical, far from it. Then you might definitely want like an image quality label for these full frame labels saying, oh, these things are bad. We don't want to try to classify these frames. There is, in the desktop side, there's a similar way to specify skip categories that you don't use in training. That's not in web yet. In web, when you just train models, it's going to train over all categories. But skip classes are something you could pass into the CLI tool.
What other addition? Aiden, something has changed since you first started working on this, and so you'll notice when you pop open the run training box now, there's a new checkbox that says use annotated frames only. And so if you choose to omit, you want to omit frames from your data set, you can just delete those full frame bounding boxes and check use annotated frames only, and those will be omitted from training. So you actually don't have to train, or you don't have to annotate everything in a data set anymore. That changed, I think, a couple of weeks ago.
Excellent. Thank you so much, Brandon. Cool. Scott, does that answer your question?
Yeah, that's great. Thanks.
Okay, perfect. It looks like we're at 11 o'clock. If it's all right with our other speakers, we can let Moritz ask his follow-up question. And then I think we'll take a 10-minute break for everybody to stretch their legs and get some water before we move on to the tutorial.
Yeah, I'll just go ahead then with my question. I'm interested in speeds a little bit. Let's assume maybe 10 hours of video with 10 objects per frame that I want to detect. How many hours would it take to run an already trained model over those 10 hours of data, given that it has around 10, let's say, objects per frame. Do you have any numbers of, kind of speed values for maybe on VIAME.com, but also on a dedicated server where somebody might have some experience?
Yeah, I mean, that the speed of the algorithms is going to depend heavily on a few factors. It's going to depend on the resolution of the imagery, what method you're training. Full frame classifiers are significantly faster than object detectors. I think we were talking about object detectors with 10 subframe objects. So there's multiple ways to run detectors too. It's going to depend on the resolution of your imagery, whether or not you have something called multi-scale turned on, which will be tried. The system will try to determine if multi-scale should be turned on by default. But say you have really big objects with respect to the frame size, that's a little bit of a different problem than detecting tons of really, really small objects, because that's going to switch the resolution input to the algorithms. So it depends on the size of your objects with respect to the full frame as well.
Let's assume small objects.
If you're looking for tons of small objects, it's going to depend on the resolution. Typically what it'll do, it'll grid up a larger resolution to like 800 by 800 tiles and process each of those tiles individually. So how many tiles there are is going to depend on the resolution of your imagery. If it's like 1080p data or 720p data or 480p data, that's going to control the processing speed. Your GPU is going to control the processing speed. How many GPUs you have in the system is going to control how fast it goes. Typically on a one mid-range consumer GPU, we've seen people go like, I don't know, 5 times longer than the video length at like 10 hertz. But that's another factor, is you can control the frame rate that you're doing processing at. So instead of 10 hertz, you can process data at 1 hertz if you want to be 2 times faster than the video length. So the frame rate that you do processing is another config parameter and that's going to depend on the problem. It's going to depend on whether or not you're doing target tracking in addition to detection. Target tracking is a separate network on top of the detectors to link the detections into tracks. That's going to affect things. So really, any question about speed is a case-by-case basis based on how much money you want to throw at GPUs to buy. People can hit real time, and we do real-time processing on some of these things. For example, some of the aerial sea lion data and Arctic seal problems, that's very high resolution data, but it's low hertz, high resolution data. So it's like 4K by 4K pixels. But we're able to hit real time by doing a combination of frame rate adjustments and multiple GPUs on the platform.
Yeah, good. Thanks.
Excellent. All right. I think that concludes the first session of our webinar today. I'm going to post my email in the chat, and I'll invite the other hosts, if they're comfortable, to share theirs as well, so that our participants can follow up with other questions if they think of them.
Yep, that's certainly fine. Feel free to email Brandon or I after the fact. We also have a mailing list for help. It's viame-web@kitware.com. I'm adding in the chat here. That CCs, the 4 or 5 major developers on the program, so that's always helpful for a faster response. But yeah, feel free to reach out if there's any other questions. I'll probably listen in the background a little bit for the rest of it today. Admittedly, I need to run and grab lunch because I'm a little late on that. So we're East Coast.
Yeah, for sure. No worries. Thank you so much, Matt and Brandon, again for joining us today. And thank you to all of our participants for sticking with us through the technical difficulties to start. And we hope to see you all in 10 minutes or so when we start the second session. Thanks.